Welcome to this visual exploration of two fascinating cellular structures, cilia and flagella. This is your cell and molecular biology course, and I'm your course instructor in your general. Although cilia and flagella may look similar under the microscope, their form and function differ in crucial ways. So let's explore into their structure, roles, and how they help cells move and also to communicate with each other. Let's begin with what are cilia and flagella. Cilia and flagella are hair-like projections that extend from the surface of the cells. They help cells move or move substances around them. These structures are found in eukaryotic cells, uh, and while prokaryotes also have flagella, they differ significantly in the structure and function. So, in this video, we will see the structure of cilia and flagella in the eukaryotic cells. So, in eukaryotes, the structure is 9 plus 2 arrangement. They are built from microtubules and surrounded by the cell's plasma membrane. These structures play vital roles in movement and sensing the environment. So in eukaryotes, it is made up of uh, microtubules in a 9 plus 2 arrangement. Now let's see the structural similarities. Both cilia and flagella share a common design known as the 9 plus 2 microtubule arrangement. So this structure includes 9 doublets of microtubules arranged around the two central ones anchored to the cells by the basal bodies. In the illustration, you can see the 9 doublets which is arranged around the two central pair of microtubules. So this is the basic structure of cilia and flagella in eukaryotic cells. Their movement is powered by dyning motor proteins. They allow them to bend and beat rhythmically. You can see the outer dyning arm and inner dyning arm in the illustration. So this is the structural similarity between cilia and flagella. Now let's see the differences in movement. Cilia beat in coordinated, that is wave-like pattern, almost like oars uh, rowing in sync. This movement sweeps fluids and particles along the cell surface, while flagella, on the other hand, move in a whip-like or undulating motion that propels the entire cell forward, which is ideal for swimming through liquid environments. You can see in the illustration the cilia and flagellum and how they move. That is the direction of motion. You can see the example paramecium, which is covered by cilia. The back and forth beating of the cilia is illustrated, while flagellum in the euglena, you can see the propeller-like motion, which is uh, the direction is different from that of the cilia. Although the structures are similar, you can see the differences in movement. You can see that paramecium which is covered by cilia. I can imagine that the cilia is something like rowing a boat, the oars of the boat, we which is together propel the organism for which is moving the surrounding environment back and forth. You can imagine that in the paramecium. Well, in Euglena, you can see the single flagellum, which is moving like a propeller that is pushing the entire organism in the liquid environment. Another key difference lies in their size and number. Cilia are typically short and numerous, covering the cell surface like a dense brush, while flagella are much longer and usually limited to one or a few per cell. These differences reflect their distinct roles in movement and fluid handling. Now let's see the functions of cilia. Cilia serve a variety of purposes. In respiratory tract, they sweep mucus and trapped particles out of the lungs. And in the female reproductive system, they help move the egg cells through the fallopian tube. Some cilia also serve sensory functions such as detecting signals in the nose or eyes. Now let's see the functions of flagella. Flagella are primarily used for propulsion. We saw earlier that cilia limit is not only with the propulsion. In higher organisms like humans, they help in um, trapping the particles and also in moving the egg cells in the fallopian tubes and also help in sensory functions. But flagella is primarily uh, used for propulsion. A common example we can see is the sperm cell, which uses a single flagellum to swim toward an egg. Some protozoans like Euglena use flagella to navigate their aquatic environments, allowing them to find food and also to escape from the predators. So this is the functions of flagella. Now let's see their malfunction. When these structures malfunction, the consequences can be serious. That is, we have seen the primary ciliary dyskinesia in the previous video, which is also caused by defective cilia, which can't clear the mucus from the lungs, leading to chronic infections. In males, the flagellar defects can cause infertility due to immobile sperm. Let's quickly compare the two. Cilia are short, numerous, and beating waves, while flagella are longer, fewer in number, and move in a whip-like pattern. Cilia mostly move substances over the cell surfaces, while flagella are designed to move the entire cells. So cilia and flagella are essential cellular structures with a shared architecture but different roles. Their function is crucial for maintaining healthy respiratory, reproductive, and sensory systems. By understanding them, we gain insights into cell biology and diseases that affect human health. So, with that, I hope you got the differences and similarities between the cilia and flagella structures. Thank you for watching. See you in the next video.